Okay. Good morning. Bon dia. Welcome to everybody to this talk. For me, it's an honor to have here in the seminar my friend Lluís Montoliu. That I would like to welcome him to this seminar.
Hello. Good morning, everyone.
Okay. So we're going to start with a short introduction of Lluís for you. Lluís Montoliu is a biologist. Actually, we study all together at the university at the same time. He's a group leader at CIBER of Rare Disease, honorary professor at the University Autónoma of Madrid, director of this Spanish node of the European Mouse Mutant Archive, and also he has a large history of studying the animal in animal models using these CRISPR-Cas9 genome editing tools. Also is president of the CSIC ethics committee. He has a large metrics, so he has published a lot of papers that you can follow. Also, he's a great communicator. Here you have a link to his intervention in a TV3 program. It was really outstanding. He has received several awards. I know that he has received more, but maybe I haven't actualized all this stuff because it has a lot of recognitions. Recently, he has led he has got the adherence of more than 150 institutions to this initiative for transparency in animal research. And finally, he's a successful writer. He has now published the third edition of this book that I would recommend you. It's Editando Genes, Recorta, Pega y Colorea. He has also been working especially in this rare disease, the albinism, but he's also a high opinion leader, and well, I can call him influencer, and with more than 23,000. I hope that today you will arrive to the 23,000 followers. I recommend you to follow him at Twitter, and later, and just to finish, I just want to thank Lluís being here with us, among us, and he's going today to talk about the genome editing in biomedicine in 2021 and beyond. And as you'll see it, he's going to talk of something that is science. As I told you, science is moving very fast. So as you will see it, most of the reference that he's going to use for his talk are related to 2020 and 2021. So this is what is happening today. So, and with any more delay, I can please, Lluís, all the audience is for you. The screen is yours.
Thank you very much, Josep, and thanks again to your Institute of Research and to this master, because I'm glad to be presenting again this update on this genome editing tool. So let me share the presentation.
Please, while doing this, I just want to ask you, the students, that please think about some questions for Lluís. You can write down the questions in the right side, as you can see the preguntas. You can upload your questions during his presentation, and then we wanna discuss it. We have a large time at the end of the seminar to discuss with Lluís. Also, you can, if you are interested, when we finish, I can upload you and make the question you can do it in Castilian, Catalan, or in any language, or in English. And well, and Lluís, please, all for you.
Thank you, Josep. So as I was telling, today I'm going to be reporting about genome editing in biomedicine in 2021 and beyond. I'm most interested to update what is going on in this genome editing revolution. because as Josep was telling you, this is a moving target. So if you compare this seminar with similar seminars that I've been delivering in previous years, you will see some differences because, of course, we know now things that we just didn't know, we just ignored just 1 or 2 years ago. So today, 1 year ago, we were celebrating this Nobel Award to these two researchers. By the way, today at 11:30 in Stockholm, we will know the awardees for the new 2021 Nobel Award for Medicine or Physiology. Last year, Emmanuel Charpentier on the left and Jennifer Doudna on the right, they both received the Nobel Award in Chemistry, and they received this because they were named for the development of a method for genome editing. This was a long expected award. It was clear that sooner or later these two researchers, they would receive this. And I want to highlight that in Stockholm, the Swedish Academy of Sciences, they underline the article A method, meaning there are several methods. And this will be the only mention I will refer to other methods of genome editing. Of course, we will be focusing the talk, I will be focusing the talk on the CRISPR-Cas tools, which is the fourth and latest, probably not the last, of these genome editing tools. But as you can see, along with the years when they appeared and they began to be used, we started with the meganucleases, then we follow by the zinc finger nucleases, the TALEN. I will be briefly referring to TALEN in a little while. And since 2013, we can use this CRISPR-Cas. So 7 years later, in 2020, they both received this Nobel Award. As you know, everything regarding CRISPR-Cas started in Spain, started in Alicante, the University of Alicante, with these microbiologists, and this is Francisco Juan Martínez Mojica. Francis Mojica, and he was interested to study some peculiar sequences that were appearing both in bacteria and in archaeas, in prokaryotes, that were organized in a very subtle manner, in a very singular manner, and these repeats, he was coining the name of CRISPR, and these repeats, together with the unique sequences of the viral genomes of the bacteriophages that they were carrying, they were the basis of an immune system, of a defense system, of a genetic basis that he was documenting in 2005. So this was one of the first observations, and later on, several other researchers, and here you have 12 of them, including these two researchers I already mentioned, Charpentier and Doudna. They, in the next 20 years, they developed all these different compounds until Charpentier and Doudna, they published in summer 2012, in June 2012, they published and they described what is called the original CRISPR-Cas9 system. And as you can see, this is a binary system originally. It is coming from a pathogenic bacteria, Streptococcus pyogenes, and it's composed of a nuclease, Cas9, that is able to do a double-strand break, this DSB, but this is not done randomly. This is done at a target, and the target is being selected, is being picked with the help of this synthetic guide RNA, or this guide RNA, which is here illustrated in blue. So basically, you only need 20 ribonucleotides that can pair Watson-Crick pairing, base pairing, with your favorite gene, and you can trigger this double-strand break. So 9 years later, today, most of us, what we use is what you see now in the cartoon. And this is a bit more elaborated cartoon. So the guide RNA is being split to the original two compounds, what is called the CRISPR RNA and what is called the tracer RNA. The CRISPR RNA is the one that carries the homology, that targets the genome, that selects the gene that you would be cutting with the help of the nuclease, with the Cas9. And the tracer RNA is pairing with the CRISPR RNA and is associating the nuclease to the site to your favorite gene. So this being a nuclease, basically, and this is a mandatory slide in all genome editing talks, as I'm sure you know this, but we need to be aware about these two different repairing pathways. As soon as we produce a double-strand break, then this is an insult to the cell, and then we have a number of proteins that will be detecting this double-strand break. Among them, for instance, P53 is one of these proteins that will readily detect this double-strand break and will trigger one of these two main repairing pathways. On the left, you have the non-homologous enjoining pathway, where by random addition and random deletion of nucleotides, you hope you will be restoring some local microhomology that will lead to the reconstitution of the physical continuity of the chromosome. Normally, because you have added or deleted nucleotides, the final result is that the gene is no longer working. So this is today the easiest way of disrupt specifically a gene. You basically target a double-strand break, and then upon repairing this double-strand break, the gene is no longer working. Some cells, particularly those cells that are actively dividing, they can repair with most favorable pathway. This would be the pathway that we would like to be using all the time. But unfortunately, this is not the default pathway. This is the so-called homology-driven repair pathway, in which in the presence of a template, in the presence of a single-strand DNA or double-strand DNA covering homology around this double-strand break, so with homology left and right to this double-strand break, you can introduce de novo sequences, this short blue fragment of DNA that wasn't there before. And because it wasn't there before, you are editing the genome, and this is properly known as the gene edition. Of course, this can be used to introduce sequences, such as introduce a mutation, but also it can be used, as we will see, as a repairing tool, because this can also be an innovative, let's say, gene therapy approach. As you know, CRISPR is being used in biology, in biotechnology, and biomedicine. Today, I'm going to be focusing only in the biomedical applications, and the biomedical applications in which there is a number of possibilities that you can use this tool. This tool is most amenable and is most versatile. It means you can adapt this tool to a variety of different applications: deleting, inserting, replacing, modifying, labeling, transcribing, targeting, activating, repressing, as you will see in a minute. Very rapidly, the easiest is to disrupt a gene. So this is kind of fundamental. This is the 101 list of items that you need to know about genome editing. If you use genome editing per se, and you don't do anything else, basically the gene you will be targeting, normally the gene will stop working. So this is very easy, and this is now being applied in cells, in animals, in plants, in all different organisms. You can duplicate these CRISPR-Cas compounds, and if you do this in cis, that means in the same DNA molecule, you will be triggering the deletion between these two different sites you will be cutting. So you will be promoting that the repairing will be accomplished between the outside ends of the DNA fragment, and basically you will be producing, with a surprisingly high probability, you will be producing this corresponding deletion. And this is something we have used a lot in the lab, because this is something that has enabled us and many others to study the non-coding genome. You know that we have 20,000 genes, but these 20,000 genes, they occupy not less about 2% of the genome. So the remaining 98% of the genome is full of repetitive sequences, is full of satellite sequences, transposons, retrotransposons, but also that's where normally you will find the regulatory elements. It was very difficult before to target these regulatory elements because they normally were surrounded by repetitive sequences, thereby inhabilitating the capacity for using standard traditional homologo recombination approaches because the presence of these repeated sequences was inhibiting the use. But now with the CRISPR, you can target a regulatory with surrounding double-strand breaks, and this is what we have done in several publications in my lab, trying to understand the regulatory elements of a gene that is involved, that is key in the pigmentation. It is an enzyme. This is tyrosinase that is encoding the first enzymatic step of the melanin biosynthetic pathway. And as you can see, you don't need to be an expert to see this gray mouse. This gray mouse is gray not because we have mutated the gene, but because we have altered one of these regulatory regions. And the fact that it's gray, and taking into account that these mutations are recessive, is that it tells you that the system is so successful that normally the animals that you are producing, they are not heterozygous, but they are homozygous. They are both carrying the mutations in both alleles. We have done this with several regulatory elements, and these are some of them, deleting these elements at 5' or 3', and this has enabled to interpret and to build this model of action of this chromatin surrounding the tyrosinase. So this is something to be underlined because for 20 years or so, we couldn't assess, we couldn't do these experiments. But now, thanks to this possibility, thanks to the CRISPR tools, we have been able to do these experiments. Of course, we can do inversions, we can promote inversions, duplications, or any other chromosomal rearrangement. We can do also point mutations, and this is something we have exploited a lot also in my lab. So reproducing human mutations that we diagnose. So we do genetic diagnosis of human rare diseases, particularly albinism, and those mutations that we found, we can transmit exactly those mutations to the corresponding mouse using the CRISPR technology, and the resulting mouse becomes the avatar. I'm using the metaphor as the science fiction movie, because remember this blue beast, they were associated, each one of them, with one human being. So I'm using this metaphor to illustrate the fact that, for instance, this person, this is a person with albinism, with one type of albinism, this person has a mutations in one gene, it's called SLC45A2, and this in this gene, she's missing a C, a single letter, a single nucleotide, a C. And the very same C is being missed in the mouse that is shown above her on the right, and this is the avatar mouse. So we cannot do experiments directly on the human being. This is, of course, understandable, and this is ethically impossible. But we can do the experiments on a similar animal model that is carrying the same mutations, and we can, for instance, validate the treatments using molecules or reposition drugs. What else we can do? We can do knock-ins. We can introduce large fragments of DNA, and we can do things that at the very beginning, when they were presented, they were already suggesting that there will be a second revolution. And this is, what about if the nuclease is inactivated and is not possible to cut the DNA? So what about if we produce an nicase? An nicase will be a Cas9 that is only cutting one of the DNA strands, or a Cas9 dead, the dead Cas9, in which the nuclease is no longer cutting DNA, but still is able to locate a given position in the genome, because thanks to the guide RNA, it will be sitting somewhere in the genome. Now, if we associate an effector domain to that nuclease, this nuclease is no longer a nuclease, but perhaps is an activator, because now we can pair this dead Cas9 with an activator, and we can promote the activation of a gene. Or we can do the opposite. We can repress a given gene specifically, and this is a specific repressor that can be triggered. This has been exploited over this year with this CRISPR on and CRISPR off, so you can really switch on or switch off. You can operate on the activity of genes using these approaches. This is quite remarkable, because before we were using such as the tetracycline system to regulate the expression of genes, but sometimes these were leaky experiments. In this case, these are very specific experiments, and using CRISPR, we can specifically reactivate, or we can silence a given gene. So what are the limitations? So far, it is a wonderful tool, but this tool has limitations, and what we can do about these limitations. So the first limitation is obvious, because if we are using an RNA molecule to target our favorite gene, this RNA molecule maybe can pair somewhere else in the genome because of the little homology. For instance, we are using a guide that has 20 ribonucleotides exactly homologous to our favorite gene, but somewhere else in the genome, there is another sequence that has 18 of these 20 that are exactly the same. Of course, the guide will not be pairing exactly 100%, but maybe it's pairing 95 or 92%, and this is enough to trigger the cutting of the Cas9, and we will be promoting these mutations or these editions not only in our favorite gene, but in a similar gene. And this is, of course, one of the big dangers. We can reduce these dangers to something below the threshold using very elaborated bioinformatic programs. A couple of years ago, we published a list of programs that you can use. At the time, there were more than 100, and now clearly the list has enlarged, and we have even produced one of these programs. We call it breaking Cas. So these are programs that you can use with your favorite genome, with the favorite species, whether you're working with human, or you're working with mice, or you're working with rats, or with macaques, or with non-human primates, or whatever. So basically, you can select the most suitable guide that is pairing only your favorite gene and nothing else. Well, this is something that you can deal with, this first problem. The second problem is a bit more complicated to deal with, which is the on-target effects, which is the mosaicism. And this is clearly because this HDR that I told you that in our dreams will be our best preferred repairing pathway, it's not operating in most cells. In most cells, such as neuronal cells that are normally not dividing, you only have the non-homologous end joining. And this is happening differently in every single cell. So in every single cell that the repairing pathway has to repair the double-strand break, it will be repaired differently. So you will be producing many different alleles. This you can see very easily when you work with pigmentation. This mouse should have been uniformly pigmented, and instead it's a mosaic. And it's a mosaic because not all cells were completely repaired. I was injecting the CRISPR-Cas tools in a one-cell embryo, but the embryo was dividing. This is a morula with 8 cell embryos, and in each cell there are 2 alleles. So 8 times 2 makes 16 possible alleles, 16 possible ways that this double-strand break can be repaired, and not all the time they will be repairing that will be along with your expectations. If you don't work with pigmentation, the way you can see this variability is by sequencing, by cloning and sequencing. And these are different mice that we produce after one of our CRISPR-Cas experiments. And as you can see, there is a lot of noise, there is a lot of variability, and there is a lot of alleles. Among them, of course, you will find the one that you are interested, but you will have to segregate it out of the rest that you are not interested. How do you do this with mice? Well, with mice, you know that all mice that are coming after a CRISPR-Cas injection, they are all mosaics. All the founders, they will be carrying several alleles. You need to sequence them all and find the one that is carrying your expected mutation. And once you find this expected mutation, you have to put it to breed, and then you have to cross it with wild-type mice. You have to expand, and you hope that these different mutations will be segregating apart, and this segregation will let you to establish a mouse colony. So this is very easy with mice, but this is, of course, a challenge with patients. You cannot treat a patient and tell the patient, we don't know what's going to be happening to you, but maybe one of your children will be inheriting a corrected gene. This doesn't work like this in a hospital setup. So this is why in the academics, the CRISPR-Cas is already de facto a revolution, whereas, as you will see in the hospital, it is coming, but it's coming step by step, and it's not coming at the full speed as we would expect. Well, there are exceptions, and the exceptions in which the non-homologous enjoining is favorable and has a benefit. And this is a work that was published 2 years ago by Paula Rio and Juan Bueren at the CMAT in Madrid, and they're studying Fanconi anemia patients. As you know, they have a problem in the hematopoietic stem cells. Fanconi anemia proteins, they are important for repairing DNA. And of course, they were promoting this non-homologous enjoining, and they were facing these mosaics. But of course, those that were goodly repaired, those that were repaired according to your expectations, those that restored the proteins, they had a proliferation advantage. And this proliferation advantage, that can be converted into a clinical application. Well, this is very rare. Normally, this mosaics is a problem, but in this case, as you can see, this mosaics can be a benefit, can be something that you can use. So what can you do today, 2021, to solve these limitations? Well, the main solution is coming from a variation in using these CRISPR-Cas tools. And the variation is called the base editors. And this will be the subject of most of the rest of my talk, because several years ago, base editors were something very sophisticated that most of us, including myself, by the way, we thought that they would be very difficult, that they would reach a clinical setup. Well, I was wrong, and I don't have any problem to admit that I was wrong, because in such a short period of time, between 2016, 2017 to now, so 4 or 5 years, they have produced many more editions, many more versions of these base editors, which are more sophisticated, which are even more optimized. And nowadays, it's probably the best genome editing tool. So what are these base editors? What is base editing? Well, base editing is composed of a nickase. This is a Cas9 that is cutting only one of the DNA strands that is associated with a chemical activity. And this chemical activity is called deaminase. This is thanks to the work of David Liu. David Liu is working at the Broad Institute in Boston. And there are 2 different flavors of these base editors, the so-called CBE and ABE. The CBE is able to convert a cytosine into a thymidine. So the C becomes a T through a uracil, whereas the ABE is a base editor that is able to convert an A into a G, an adenine into a guanine, so through an inosine. So basically what it does is that they promote chemically the conversion of the nitrogenated base, the conversion of a nucleotide without double-strand breaks. And because you are not producing double-strand breaks, you are not generating, you are not triggering mosaicism, and you have also a very low level of these off-target effects because of these new versions. So this has been continued, but essentially this is like a corrector. This is like a typographic corrector. We erase the letter that we have to convert, in this case a C, and we put a T in the same place. So this is an ideal. This is actually a miracle. This is a tool that basically is now producing already the first successful experiments. Recently, this year, we had a 3rd flavor. So we had A to G, we have C to T, And now we have a new base editor, of course, also coming from David Liu lab, that is converting a C into a G. This is how it's been mediated. This is a number of different chemical reactions that will convert this C into a G. So how this is being seen in reality? Well, in reality, the problem is that you need to know very well the size, the atomic size of the protein, because you have to calculate the guide so that the Cas9 associated with the chemical converting domain, with the deaminase, is touching exactly on the nucleotide that you would like to update. So this seems easy, but it's nothing but easy. So basically, this is a very difficult, and this was very challenging. And this was something that many of us, we consider this will be a limitation, a great limitation for these base editors. For instance, this is a mouse model of hereditary tyrosinemia type 1, and basically the A you would like to change is this A at position 9, and you want to convert it into a G. And in addition to converting this A into G, you are also seemingly converting A7, A5, and A3. It means it is okay, it is producing the expected modification, but it is also converting other A's that are in the vicinity. And that was the risk, and that was the limitation in the first versions of the base editors. This was a paper published in 2020, so you will be surprised that in less than a year, the base editors have evolved very rapidly. And for instance, beginning of this year, David Liu surprised us all with this mouse model of progeria. This is the Hutchinson-Gilford progeria. This is an ultra-rare disease. You know, this aging, this accelerated aging, such as this kid, in which they normally die in their youth. And this is all mediated by a mutation in the lamin A. This is one of the nuclear proteins that is most important for gene expression. And this mutation can be restored with the C to T conversion. So the CBE is the one that can be used to convert. And this is actually what they did. They did this in cells and in mice. And this in vivo based editing was extending the life expectation of these mice from around 200 days to around 500 days. So basically, they more than double the life expectancy of these mice. So definitely, they fixed the problem in most of the cells of the body. They also tested before in cells, and they verified that in most of the cases, they targeted the corrected C, and they converted into the corresponding mutation. So other examples that we have known this year, they also refer to mouse models of, for instance, sickle cell disease. Remember, sickle cell is mediated by a mutation in the beta globin counterpart of the hemoglobin. And these beta globin mutations, you will see later on that another approach is to substitute the beta globin by a gamma globin, so reactivating gamma globin that is normally expressed during the fetal development. But in this case, they decided to convert this mutated valin, this as you see in the upper part of the slide, they converted into allele that is called Makassar. And this Makassar allele, it's already existing in the population. It's not the wild type allele, but it is an okay allele. It is producing a beta globin that perhaps is not functioning 100%, but is functioning very well. It means we can use base editing to convert this mutant valin into something that is an amino acid that exists already in the human population and is producing a functional hemoglobin. So again, this is solving this problem through base editing. It means we are no longer using CRISPR-Cas to cut and promote this random repairing that is producing many different solutions, which normally they are not the ones that we are expecting. In this case, we are selecting exactly which nucleotide we would like to update, and we apply these chemical modifications, and the result is these mice, which in this case, they are a sickle cell mouse model that is converted to an almost wild-type mouse model. This is not only applicable to rare disease, because we have this thought, and you will see that most of these applications that have reached the clinical setting, they refer to rare disease of genetic basis. But what about a very common disease? What about targeting cardiovascular disease? What about trying to lower the content of the cholesterol content that we have in our blood? As you know, 30% of people that will be facing any cardiovascular disease that is normally triggered by the high cholesterol levels that we have in the blood. So this is not a rare disease. This is a very common setup. Well, in this case, again, this Kiran Musunuru and other collaborators, they decide to use base editing, in this case, not to change a gene, but to kill a gene. So base editing can also be used to inactivate a gene precisely. And they choose to inactivate the PCSK9. PCSK9 is a normal repressor of the low-density lipoprotein receptor. It means when you have high levels of PCSK9, you will have low levels of the receptor. This is normally being expressed in the cells, in the liver cells, in the hepatocytes. You will have a very low uptake of these lipoproteins that are in the bloodstream. But if you inactivate the PCSK9, basically what you are promoting is an overexpression of the receptor. And this overexpression leads to an increase of the uptake of the circulating LDLs. And as a result, you reduce the circulating LDLs and you reduce the cholesterol levels in blood, and thereby you also reducing the risk of atherosclerosis and cardiovascular diseases. So as you can see, using base editors, and this is, I think, an extraordinary work indicating and showing the way how this can be converted sooner than later into a clinical application. By the way, this was confirmed not only in mice, but in non-human primates. So I'm sure that this will be converted into a clinical trial very soon, and we might be observing some of these benefits into the human population. By the way, inhibitors of the PCSK9, they are already applied, they are already used in the clinical setup, but in this case, we will not be using inhibitors, we will be targeting, we will be killing this gene, and thereby we will be producing this uptake of this LDL. So what else has been available using base editors recently, and normally always associated with David Liu? Well, this is not an ultra-rare disease, this is not a very common disease, but this is something in the middle. This is the commonest of the rare diseases. Unfortunately, as you know, this is cystic fibrosis. Cystic fibrosis that is normally with mutations associated with the CFTR. And in this case, they apply base editing with cells. These are human airway epithelial cells, and they target a very common mutations, which is this delta phenylalanine 508, which is the commonest mutation in cystic fibrosis. And they apply, in this case, these adenine base editors to restore the correct sequence at a very high efficiency. More than 80% of the cells, they got the mutation corrected. So definitely with the newest versions of the base editors, you really can target and you really can promote. So base editors, they also have been used to solve a long-standing problem that was solved last year. And this is about genome editing the mitochondrial DNA. You know, mitochondria, They are derived from ancient prokaryotes that were decided to live together with other prokaryotes, and they form the eukaryotic cell. So this is why mitochondrials, they still remain and have some DNA. And you know that there is a very difficult to handle, very difficult to deal with mitochondrial-related disease that are associated with mutations in the mitochondrial DNA. Well, the problem of using CRISPR-Cas with mitochondrial DNA is we know how to import proteins into the mitochondria, but we don't know how to import RNA. And since CRISPR-Cas, they require an RNA guide, it is very difficult, and so far it was a limitation to use CRISPR-Cas in the mitochondria. So David Liu, they converted, and this is, as I told you before, they rescued the TALEN, and the TALEN is a genome editing tool that came shortly before the CRISPR-Cas. The TALEN is using proteins to target a given DNA sequence, so they are not using RNA. And they associated the TALEN with this cytidine deaminase, which is similar to this deaminase activity that is being used in the base editors. So basically, they created a base editor for the mitochondrial DNA. And this C to T conversions that is promoted with these talents is now enabled and basically now permits the updating of the mitochondrial DNA, which has or might have some therapeutical consequences. There is a second version of the base editing, which is not properly saying a base editing, but is called the prime editing. The prime editing, prime like quality. This is first quality editing. And this was coming from the imagination again from David Liu. And David Liu was imagining the association this time of a nickase with a reverse transcriptase. And the single RNA was extended in the 3' N, and the 3' N is now associated with the opposite DNA strand, as you can see here in the drawing. So because you are associating the nickase with a reverse transcriptase that is able to synthesize DNA using RNA as a template, basically you're extending the DNA, you're extending this complementary DNA strand, and you can go over the mutation and you can fix a mutation. And this is very precise, and what is more important, this is not associated with these off-target or mosaic effects. One of these first examples has been published in Mice this year, and this is a side-by-side comparison between standard CRISPR-Cas9. Well, we say standard, it's just 7-8 years ago that we started using CRISPR-Cas9, but we can call already this a classical CRISPR-Cas9 versus prime editing. On the left, you have standard Cas9, and basically you get a very good percentage of correct on targeting. This is 56%, but unfortunately you get this very high mosaicism. You look at this, what happens on the right. On the right is prime editing. You have a reasonably high correction of on targeting, about 21%. This is also very high, but what is more important, you no longer have mosaicism. And this is something to be underlined. And this is something that is important to be taken into account. Both base editing and prime editing, they are not associated with mosaicism, and they are associated, the last versions, with very low level of off-target effects. So where can you read about this the new generation of the CRISPR-Cas tools? Well, there are two very good reviews that I'm recommending you. This appeared last year in Nature Biotechnology by David Liu, where he described these different base editors and prime editors. And very recently, it appeared last week in Molecular Therapy. Actually, it corresponds to the November issue of the Molecular Therapy. David Liu has prepared this updated review that I'm highly recommending, that is comparing CRISPR-Cas9 editors with the base editors and the prime editors. So this is probably the latest review that you can use. So what about biomedical applications? How can we apply this into the clinical setup? Well, the normal application would be the gene therapy approaches. Gene therapy approaches, because if you think it carefully, this is the picture we have all in mind. So ideally, we thought, we know there is a gene with a given mutation is no longer working. We would like to cut with a traditional CRISPR-Cas9. We would like to cut very near this mutation. We will be providing a template DNA with the correct sequence, and we will be triggering the conversion of this mutation into the correct sequence. So this is the ideal gene therapy now that we can use with CRISPR-Cas9. But this is very similar to what has been applied since the very beginning of the gene therapy approaches, both in vivo and ex vivo. In vivo, treating directly the patient; ex vivo, first collecting cells from the patient, kind of genetically modifying these cells in the lab and returning the cells to the patient. So this is the traditional gene therapy approach that we know for more than 30 years. In the late 80s, beginning of 90s, this was what was hoped. So we have a mutation, and we correct the mutation, and we convert the mutation in the correct gene. How can we do this? Well, over the years, we have been doing this in very different manners. We can substitute a small piece of the gene that carries the mutation. We can substitute the entire gene. This is normally very challenging. This is only possible where we can know the homologue recombination tools, or we can add the gene, the therapeutical gene, somewhere else in the genome. This is what was feasible, and this was very familiar and very popular a couple of years ago. For instance, you remember, and it's being used in the clinical setups, these lentiviral genomes. Using lentiviruses, you can promote the reintroduction of a functional copy of your gene, but you don't touch the mutated gene. The mutated gene remains. You are only providing an additional copy somewhere else. Well, this somewhere else has some problems, or might have some dangerous consequences, because you might be landing over another gene, and you might be inactivating another gene. And as you know, using retroviral genomes and using lentivirals, the first generation of lentivirus, they were associated with some leukemias, because you not only were correcting the gene you wanted to correct, but you will be reactivating oncogenes. So this is bad news. So this is why these previous gene therapy setups, they are no longer being used, and mostly they are not being used, and they have been substituted by these episomal treatments. So this is providing a new gene instead of somewhere else in the genome, providing it as an episome. And the best episomes that we know of these days are the adeno-associated viral genomes. So the adeno-associated viruses, this AAV, they are used to deliver these functional copies of the gene. And again, we are not correcting the gene, we are just providing a new copy of the gene that is now no longer interrupting the genome. This is currently the standard in gene therapy, and this is probably one of the first products that you might have seen recently being approved in Europe. It was first approved in the States. This is Luxturna, that was originally produced by Spark Therapeutics, and it's including a gene that's called RPE65. It's a gene that is used for treating retinal degenerative disease, a number of these different diseases in which this gene is being involved. So you can deliver directly this gene to the appropriate retinal cells through an intraocular subretinal injection. In this case, these AAVs, they will be delivering this genome. They carry this CRISPR-Cas, not CRISPR-Cas tools, this normal gene, this RPE65 gene, and this will be used for therapy. So how can we convert CRISPR-Cas now that we know what is being occurring today with gene therapy? Well, the reality the reality with CRISPR-Cas currently, and this we didn't know some years ago, is that we still don't know how to substitute a mutated gene into a correct gene. So triggering this homologous-driven repair pathway, the right pathway, as I showed you before, it's still very challenging. It's still very difficult. So this is why most of the applications that you will see, they have been appearing in the clinic, they are used to target the mutated gene. So you inactivate the gene through CRISPR-Cas. This is probably a very good idea because CRISPR-Cas, it's a very specific, and this is what it's meant for. CRISPR-Cas originally was a defense system used to target the viral genomes of bacteriophages infecting bacteria. So basically, we are reusing the CRISPR-Cas using the original property. So we are targeting this mutated DNA, and we are inactivating this mutated DNA. And this has some therapeutical consequences. Examples. In 2016, 5 years ago, the first experiments that were leading to a somatic gene therapy, this was in a mouse model in mice, they were with a mouse model of Duchenne muscular dystrophy. In this case, the dystrophin gene was carrying a given mutation in a given exon, and this exon was removed. So again, we were using the targeting, the cutting effect of this Cas9. We cut before and after the mutation, and we promote the excision of this deleted exon. So nowadays, if you check the World database of human clinical trials, there are, well, this was last week, so I assume, I haven't checked again, but I assume this is still the case. It's 50 clinical trials today that they are using CRISPR, and most of them are using an ex vivo. So again, most of them, they are using cells that have been obtained from the patient that can be edited in the laboratory. Of course, this can only be applied to blood cells or to skin cells, those cells that you can culture, you can cultivate outside the patient, and you can modify it before returning and have a therapeutical effect. So this is mostly true for the immunotherapy of cancer. So this is becoming more and more useful. I'm sure in this hospital, in the Hebron hospital, the CAR T cells is a very popular or relatively popular therapy. And as you know, the this CRISPR can be used during this CAR T preparation, because with people that are experiencing very aggressive cancers, such as myeloma, sarcoma, or melanoma, these are patients that unfortunately are no longer responding to chemotherapy or to radiotherapy. You can extract T cells, T lymphocytes from these patients, and you can apply CRISPR to inactivate both the PD-1. PD-1 is a negative regulator of the immune system. So if you remove PD-1, the cells are strongly reacting against the cancer cells. You might have maybe an autoimmune effect, but this is the list of your problems when you are dealing with one of these aggressive cancers. But you can also use CRISPR to inactivate the T cell receptor. And upon doing this, you are producing a T lymphocytes that are more aggressive, and they do not have a T cell receptor. But then before returning these cells into the body of these patients, you are infecting them with a lentiviral vector that is carrying a chimeric T cell receptor, that is targeting specifically some membrane antigens that are being exposed in these cancer cells. So this is a treatment that unfortunately is not always working, but in 30% of the cases, maybe 40% of the patients that would die if we wouldn't do anything, because there is no solution, there is no treatment for these patients. In this case, with the CAR T cells, some of these patients, and I hope that more and more patients will be successfully adding to this list, some of these patients will be survived. So this is an ex vivo use of CRISPR. This started in China in 2016, and this is now being used in many different hospitals, also in this country. Another ex vivo therapy, most successful, is, as I told you, it's a treatment for both sickle cell anemia and beta thalassemia. So, beta globin is mutated, and in this case, instead of correcting beta, what these investigators and these researchers, they decided is to reactivate the gamma globin, which is a similar gene that is normally being expressed during fetal development. Normally, this gene is no longer working when you are in the adulthood because you have a repressor, and this repressor is called BCL11A. And these investigators, they envisage using CRISPR to inactivate one of the enhancers of this receptor. They could not kill the gene because this BCL11A, it's been required for several steps during the blood hematopoiesis. But if they target this enhancer with CRISPR, they remove the enhancer, similar to what we do in the non-coding genome with our mice. They reduce significantly the levels of expression of this repressor, and without this repressor, then you have blood cells that are expressing gamma, and the gamma globin can substitute the beta globin. And this is something very simple, a very neat idea that was put into practice with Victoria Gray. This is the first patient that was treated in July 2019, and 18 months later, in December 2020, in New England Journal of Medicine, you see the green bars that are being substituted by the blue bars. The green is the beta globin expression, and the blue is the gamma globin expression. I would like to stress that this is a single treatment of these women that was subjected to weekly blood transfusions and no longer requires blood transfusions because their blood is already being corrected. This is the first case of a cure that is mediated using CRISPR-Cas. There is another approach that has been used in vivo. I told you that most of the CRISPR clinical trials nowadays are being applied ex vivo, but there are two examples of an in vivo. The first one is something that started end of 2018, beginning of 2019, and that was with the aim of correcting a mutation in the CAP290 gene that is causing the retinal degeneration disease that is called liver congenital amaurosis type 10. In this case, these were the companies associated. Again, this was using adeno-associated viral vectors, intraocularly and subretinally delivering these CRISPR-Cas tools. But the aim is to eliminate a cryptic signal in one of the introns that was responsible for the aberrant processing of the RNA. Upon using two CRISPR-Cas compounds, they would be targeting this molecule in cis, and they will be promoting a repair, and the resulting intron, they would be normally spliced. Well, probably in the last week, you have heard about this news. And some of these news are a bit exaggerating the results. They say they return people that they could not see, now they can see. Well, this is not exactly true. So this is the first release of the results of this ongoing clinical trial in which it's phase 1, phase 2. It's about safety. So they say that the doses that they have been used, they don't have apparently adverse effects. And they have, look what they say, they are very prudent. They have efficacy signals in the mid-dose cohort, provide initial support for clinical benefits. Well, this is a very weak, and this is, even though positive, this is a very prudent way of saying it seems it might be working. But we have to be prudent, and we have to wait and see until these results, they are appearing in a peer review journal. So These approaches, as I told you, they were mostly using these adeno-associated viral vectors, but very readily we got to know as an alternative approach this for treatment, for the treating of rare disease. In this case, it's a disease that is called Transthiritine Amyloidosis. It's a congenital disease. It's associated with the TTR gene, and the overexpression of this protein is fatal. So basically, it produces the death of the patients because it targets mostly muscle cells and neuronal cells. You have cardiomyopathy, you have neuropathies, and basically what these investigators associated with this company they did, they were including inside lipid nanoparticles, they were including RNAs that were carrying these CRISPR-Cas tools. So basically, they were targeting the gene, again, not aiming to correct a mutation, but to silence the gene that will have a therapeutic effect. Well, this has been updated very recently this summer, and this is a New England Journal of Medicine article in which they show the results not only in mice that were already published before, but also in non-human primates, and the first results of a clinical trial with human beings. Again, this is a lipid nanoparticle that is carrying the RNA that encodes this Cas9 and the RNA guide, and the Cas9 is targeting the TTR gene. So basically, this is the product, this is the drug they use, and they also combining these lipid nanoparticles with ApoE, and they are using ApoE because they want these particles to be captured by the hepatocytes where this gene TTR is normally expressed. These are the results in macaques. These are the results in non-human primates in different doses. And as you can see, and these are the lower graphs, as soon as you provide these lipid nanoparticles, you reduce the level of the TTR in the bloodstream. And these are the results. This is no longer mice or macaques or primates. These are human patients. These are only 6 patients, 3 patients with 0.1 milligrams per kilo, and 3 other patients with doses that is three times higher. And in both cases, they see in just a month, this is a month treatment. Of course, the experiment will continue. And after a single administration, they have a significant reduction. And this is important because I would like to finish my talk, but just reminding that sometimes technologies appear and they fuse, they merge. And as you have seen, this last experiment is using these lipid nanoparticles and RNA. And what are these technologies? Well, these are exactly the same technologies that we have become aware with the Pfizer-BioNTech and the Moderna vaccines against COVID-19 that were approved at the end of December, that they have produced so many benefits and they have reduced significantly the number of infected people and then particularly the number of dead people. This is the basis of these vaccines, of the COVID-19 vaccines, lipid nanoparticles that are transmitting RNA into the cells. RNA that normally wasn't used as a therapeutic agent because RNA is a very fragile and can be not only degraded, but also RNA, it's normally interpreted by the cell as a viral genome, and this is triggering a response. This is triggering an immune response. This is why most of the vaccines that we have today are based on proteins or are based on DNA. For instance, the AstraZeneca or the Janssen vaccines that are based on adenoviral genomes, they are DNA vaccines. The protein vaccines, such as many other recombinant proteins that are used in vaccines, these are the most popular, whereas RNA was normally forgotten. And that was because people didn't know how to deal with the RNA, how to stabilize the RNA. And this was the work done by these two investigators, Katalin Karikó from Hungary, later on developing her work in the States, and Drew Weissman. Drew Weissman was working with Anthony Fauci in the NIH in Bethesda, but later on they both met in Philadelphia at the University of Pennsylvania. This is where they published this seminal paper in 2005. In 2005, they realized they could substitute in the RNAs the uridines, the U's, by pseudouridines. This is almost chemically identical with a small variation, but just this small variation was enough to stop triggering an immune response. And they could verify that by using this, they had lower levels of TNF-alpha, lower levels of different interleukins, such as IL interleukin 12, and etc. So that was the basis of using RNA as a therapeutical agent. And later on, this was Drew Weissman in collaboration with many other researchers, that they decided to embed these RNA molecules, these messenger RNA molecules, inside these lipid nanoparticles. That was 10 years later. This was another Karikó and Weissman paper, and this is in 2015, where they document that encapsulating mRNA with these lipid nanoparticles will be extending the translation period, and they checked different routes of administration. Well, these are the fundamental papers, these are the fundamental publications that were possibilitating not only the production of the vaccines, but also the production of alternative routes of administering the CRISPR-Cas tools. And as you can see, here we have a confluential of different technologies. We are mixing up CRISPR-Cas with RNA technology, with nanotechnology, with lipid nanoparticles. And this is basically what we will be having today. So today, what is likely going to be happening, not only in academy, but also in clinical trials, the CRISPR tools are used to inactivate genes. So we are not using the traditional Cas9 to correct genes, because we know that this homologous recombination pathway is very challenging. We still don't know how to monitor, how to control this pathway. So we're using CRISPR-Cas9 standard for inactivating genes, because they know what they do. This is exactly what they're good for. What can we use to correct genes? Well, we can use another variant of the CRISPR-Cas, which are the base editors. And we are also combining with two other technologies, which are the messenger RNA and the lipid nanoparticles. Finally, to tell you that the list of Cas nucleases is enlarging. 50% of bacteria and 80% of archaeas, they have a CRISPR-Cas system. Well, this is the latest summary of CRISPR-Cas system that is published by a number of key people, including Francis Mojica and many others in the CRISPR-Cas field. And basically, we have two classes of CRISPR-Cas systems and up to 6-7 types of CRISPR-Cas. So there are many CRISPR-Cas systems that we can explore, that we can follow up, and that we can use, including, and this is my last slide, this is the grandmothers of the CRISPR-Cas that were very recently published by Feng Zhang. Feng Zhang is another key player in the CRISPR-Cas field, and very recently, in collaboration with Eugene Koonin from the NCBI, they discover that some transposons, they are carrying some molecules that are very tiny Cas-like nucleases, that probably they are the origin, the evolutionary origin of the CRISPR-Cas systems. Well, I finish here, and I'm going to be thanking all the different institutions that are providing us some money for doing our different experiments. And I'm thanking all these different people that have been with us over these very difficult 1.5 past years, in which we had to continue doing these research experiments together with the pandemic. And I'm sure this was happening in everywhere, also in your institutions. I will take any questions right now. Thank you.
Okay, thank you very much. Lluís?
I think I've been more than just 1 hour and 5 minutes, but I think should be okay. Yeah?
Yes, thank you very much. Will be perfect. And so we have enough time now for discussion. So we're open for questions. So please, to the audience, don't be shy. It is your opportunity to discuss.
We have already.
Yeah, we have some here, but please coming with many other questions.
So I will be responding in the same language they use, okay?
Yes.
Yes. Aida diu, bones, amb aquesta tecnologia podem ser al·lels específics, és a dir, si una malaltia X és causada per una mutació en un dels dos al·lels, la tecnologia de CRISPR per fer modificacions específicament a l'al·lel mutat sense afectar l'altre, justament, Aida, justament. Això no sempre...
Lluís, if you can do it in English or Spanish, because in the master we have people from all over Spain.
Okay, so basically, I'll do it in English then. So yes. Basically, Aida is asking whether we can do allele-specific targeting using the CRISPR-Cas, and I was saying yes, of course. It certainly depends on the sequence. Not all sequences are possible because you need to choose your guide that is targeting specifically your allele and it's not targeting the wild type. Sometimes it's possible with some nucleases, some other time you need to use another nucleases. So this is why we need different nucleases. The more nucleases we have, the more different guides that we can use and the more different pump signals that we have to take into account, either at the 5' or at the 3' end. So in short, basically we can apply this for allele specific, and this is already being used. So Alejandro says, imagine that you want to create a knockout, let's call it of a given gene. Supposing that the gene is repaired by non-homologous joining, but it's possible that the repair creates a change that is in frame and the modified protein continues to be expressed. Well, this is not something that I have to imagine. This is something that is happening all the time. So basically, this is the result of the non-homologous joining, that not all productive results are also ending the way you plan. So basically, if you're using a CRISPR-Cas setting up for inactivating a gene, it might be that some of this mosaic variability of the alleles, some of these alleles might still recover some function. But what you expect, what you hope, is that most alleles will be inactivating alleles. So this is why, if you look carefully and you read this TTR paper, this last paper that was published in June in New England Journal of Medicine for this amyloidosis transthiritine, you will see that the inactivation is not 100%. The inactivation sometimes is 60, 70, 80%, meaning that first, there are cells that they have not received this liquid nanoparticle. This can be, so you are not targeting all the hepatocytes. And second, you might be targeting an hepatocyte, but you might not be inactivating that corresponding gene. This is why you don't have 100%. But sometimes you don't need 100% in order to get a therapeutic benefit. And this is this window of opportunity that that is being used in clinical setup, because you have this threshold. You need to go below a given threshold to be therapeutically effective. So Maria Viñas, he says, or she says, I don't get how it's been improved, the base editors in the last 2021 examples, and how the limitation of non-modifying nearby nucleotides has been overcome. As the figures of these papers, there was still this problem, isn't it? Well, it is partially true, Maria, but if you go, what happens is that in these last 2 years, in 2020 and 2021, there has been an evolution of this CBE and this ABE, the cytosine-based editors and these adenine-based editors. So we have the latest versions are much more specific, and they trigger less mosaicism, and they trigger less of target effects, and they are likely modifying less nucleotides in the vicinity. In some of these setups, they are choosing their modifications such in a way that even in the rare case that they might be modifying some nucleotides in the vicinity, the resulting mutation is a silent mutation, and it's not affecting, it's not converting the protein. Of course, this is something that you cannot expect in all different genes. You have to check, you have to double check, and you have to validate, but this is how this is being progressed. Most of this is not released for public use. A good number of these editors, they are already, you can ask them through Agin, but some of them, they are not available to everyone. So because they are being developed by a company that is Beam Therapeutics, and Beam Therapeutics is a company that was founded by Keith Young, by David Liu, and by Feng Zhang. And this is the company that is producing the best base editors. And we just hope that the best base editors are eventually distributed, at least for academic use. Of course, for clinical use, you will have to set up an agreement, a non-exclusive agreement with this company. So this is, let's see.
Yes, we have now 3. Adria Roca.
Oh yes, they're coming. Okay.
Yeah, there are 3 more. You can order, you can order the questions. If not,
No, it's okay. It's okay. I can read this. Adria,
Jordi, and Irina.
Okay. Adria says, it's not clear to me how you can repair mutation in the mitochondrial DNA with TALENs. What is the advantage of using TALENs instead of CRISPR for mitochondrial DNA? Well, it's very obvious. Perhaps I wasn't clear enough, but I'm taking the opportunity to be more clear, hopefully now. The problem with mitochondria is that you cannot import RNA. So you cannot introduce RNA molecules inside the mitochondria. You can introduce proteins. You have transporters that they can transport proteins, but they cannot transport RNA. And because CRISPR-Cas requires an RNA guide, then it's a very obvious limitation for preventing the use of the CRISPR-Cas inside the mitochondria. So this is why David Liu decided to use another genome editing tool, and that was the previous TALEN. And the TALEN, and this is in contrast to CRISPR-Cas, the TALEN is not using RNA for guiding the nuclease to cut. The nuclease is being guided by another protein domain, a protein domain that you can produce synthetically in the lab. And according to the different amino acids that you produce, you will be targeting a different sequence. This is it's more difficult to produce this genome editor. With the CRISPR-Cas, we know that you change the RNA guide and you are targeting a different gene. With a TALEN, you need to change the protein, because according to the amino acid protein, you are targeting a different gene. Well, they use this TALEN to direct to a given sequence into the DNA, in the mitochondrial DNA. And later on, basically, they remove the nuclease domain and they substitute by this deaminase domain. So basically, they convert a C into a T, but now inside the mitochondria. So that's basically the advantage. So Jordi, I'd like to know if there is any approximation of CRISPR deactivation once the change is done to prevent non-specific modification. Well, this is a very important question, and there are a number of tricks that you can use. For instance, I haven't commented because this is kind of the technicalities of the CRISPR-Cas, but you know that the guide needs to be next to a given signal that is being used by the CRISPR-Cas system. This is what we call this protospace adjacent motif, the PAM. Actually, this was named also by Francis Mojica. And this PAM is what the bacteria are using to discriminate between the viral genomes and their own genome. Because otherwise, without the PAM, they will be cutting their own genome, because in their own genome, they have pieces of this viral genome. And they know that they are dealing with a viral genome because next to this target sequence, there is always a signal. So it means when the bacteria is collecting fragments from the viral genome, is collecting fragments that are associated with a PAM. And this is why it's a biological limitation. We need to use this pump, and we need to choose these guides that are next to the pump that corresponds to the nuclease that we will be using. One trick that we can use to prevent the further modification is that we can give a template DNA that is not only altering the sequence that we want, that it's not only introducing the mutation that we want, but it is also mutating the pump. So if we are mutating the pump, once the mutation that we want is introduced in the genome, then the pump is no longer there, and then the CRISPR-Cas can no longer cut. So this is the obvious trick that we can use. There are other tricks that can be used. There are anti-CRISP proteins. There are some phages, some viral. I mean, pretty much everything is invented by bacteria or by the bacteriophages, and there are some bacteriophages that they are sort of confronting, they're fighting against the CRISPR-Cas system with some proteins that are inhibiting the Cas9 nucleases. And this is something that you can use as well. Irina, yeah. So in the future, you can cure the albinism or similar diseases with CRISPR, or it is still very difficult or complicated because of ethical reasons? Well, this is another very good question. So there is a threshold between what is a severe disease, a life-threatening disease, and something that is not compromising your life. People with albinism, they don't die of albinism. Well, they do in Africa, because in Africa they cannot protect themselves from the sun, and because the absence of pigmentation, they led us to develop skin cancer. And the skin cancer is not being treated, and they die about a metastasis associated from this skin cancer. But this is totally gratuitous. This is totally unnecessary. In general, a person with albinism will not die of albinism. But in all people with albinism, they have a very severe visual deficit. So the question that you're probably asking is, are we able to restore at least partially this visual deficit? Well, the answer is this is what we're trying. This is what we have been investigating over 30 years in my lab. So basically, the aim, we are using animal models for it, is to restore the activity of the genes that are involved in albinism. There are more than 22 genes that are associated with mutations in different types of albinism. And the idea behind is to be able to restore the activity of these genes where they are needed. And they are particularly needed in retina development. So this is still far from being converted into an application, but possibly sometime in the future, we will be able to offer some therapies that will perhaps not cure, such as the example I was giving with Victoria Gray, but perhaps they will alleviate the suffering on this rare disease. So
you have Juan Carlos.
Juan Carlos, thank you, Josep. So Juan Carlos, so there are immune response or intracellular direct response against these CRISPR-Cas molecules or RNAs. Well, with the RNA, the answer is Carico and Weissman. The answer is converting uridine in pseudo-uridine. And this is the answer that now we have validated in millions of people that we are carrying. Myself, I got a Moderna shot. So basically, most of us we used to carry these RNA molecules that were stabilized and were subjected to different rounds of translation. So you can stabilize the RNA, and you for fighting against the molecules, well, it depends where's the origin of the Cas nuclease. This time I didn't refer because I didn't have the time, but a couple of years ago, Matt Porteous in Stanford, he was the first realizing that if we're using Cas9 from Streptococcus pyogenes, perhaps our immune system has already antibodies and lymphocytes against the Cas9, because pyogenes is causing otitis, is causing infections in the throat. So basically, it's known to our immune system, and all the compounds of the bacteria, they are also known to our immune system. And he was verifying that between 60 to 70% of people in the human population, they carry these these antibodies. Therefore, if you would be treating these patients, if these people are patients of a given disease of genetic basis, and if you would be treating these patients with CRISPR-Cas9 from Pyogenes, you will be triggering an anaphylactic response. You will be reactivating an immune response, which can be compromising the life of the patient. So the alternatives are to combine these treatments with immunomodulators, such as the one that are given to people that is being organ transplanted. So you can have immunomodulators that reduce the immune response, or better, as I told you in my last set of slides, you can explore the use of different nucleases. Different nucleases, they are not coming from bacteria that are pathogenic to human beings. So
Madeleine, you have Madeleine.
So are they using nowadays other Cas or similar proteins for genome editing, while only focusing on Cas9? Well, Cas9 is a generic name. So there are many Cas9 from many different bacteria and archaeas. So basically what you're probably asking is, what is the source of Cas9? So in the academic setup, most of us, we still use the Cas9 from Streptococcus pyogenes. In the therapeutical first setups, it was substituted, the Cas9 from Pyogenes, by the Cas9 from Staphylococcus aureus, but that Staphylococcus aureus is another pathogen that is having the same problems that I was just describing to your colleague. So nowadays we have Cas9s, and we have Cas12s, and we have Cas13s, and we have a number of different Cas. Actually, there are hundreds of Cas that are now being proposed. Now they need to be validated, and eventually they will be used. They will be used in the clinical setup. So there are many more Cas, not only Cas9s being used. I think it's for Carlos, no?
Yeah, one question by David, that it was the one that I had also for you.
Okay, David. So David Camello. Perhaps this question is off topic. No, there is no questions off topic. All questions are welcome. But CRISPR seems to be a very good antiviral mechanism for bacteria, and taking into account that the system can be used on humans for genome editing. Has anyone tried to use CRISPR as an antiviral? Well, of course, David. Well, I had to take things outside. I mean, this time, I think last year I was discussing this opportunity, but this year I wanted to focus on base editing, which I think is the novelty. Last year, the novelty was exactly what you asked, and this is something that we're doing in the lab. And there are many different Cas, including some Cas proteins that are not targeting DNA, but they are targeting RNA. And these are the Cas13 family. And we have different subtypes of the Cas13. We have Cas13A, Cas13V, Cas13C, Cas13D. There are different Cas13 that they can be used to target specifically an RNA molecule. For instance, the pandemic COVID-19 is caused by a coronavirus, and the coronavirus SARS-CoV-2, the genome is an RNA. So you can say, if we have a virus whose genome is an RNA, and we have a nuclease that is targeting RNA, why don't we use an RNA guide to target some of the internal parts of the genome? We open the genome and we produce open free ends, 5' and 3' ends of the RNA, that they can be targeted by RNases, and they can reduce the viral load. Well, this has been checked already in cells. This has been done in cells that was published in April, May 2020 by a team in Cell in Stanford. And well, they used an approach that possibly is not going to be applicable in clinics. They use an approach based on first transforming cells in culture with a Cas13D expressing cassette. So they produced transgenic cells that were overproducing Cas13D, and later on they infected the cells with RNA viruses. They use SARS-CoV-2 and they use influenza. You know that the influenza virus also has RNA. They have 8 molecules of RNA in their genome, and they also combine this infection with guide RNAs that they will be targeting internal parts of these viruses. And they demonstrated that they could reduce significantly the load of RNA. In vivo, this has been validated early this year in hamsters and in mice with both influenza and with SARS-CoV-2. You know, for SARS-CoV-2, you cannot use wild-type mice. You need to use transgenic mice that are overexpressing the human ACE2 receptor. That's the one that is being used by SARS-CoV-2. So they demonstrated that they could provide these CRISPR tools as RNA molecules together with lipid nanoparticles and that are administered through the airways intranasally. And they demonstrated a significant reduction of the viral loads of the viral titles in the influenza and SARS-CoV-2. So in short, yes, you can apply CRISPR-Cas to an antiviral approach, but the problem nowadays, with the exception of this single paper that was published early this year, the problem is how to deliver this RNA to the target cells. And for instance, our project is not using the total RNA, is using a ribonucleoprotein, is using the protein per se that is encapsulated in some nanoparticle, and that is also including the RNA. Perhaps the solution will be to convert everything into RNA molecules, use the same vaccine COVID-19 technology from Carico and Weizmann, and be able to apply this antiviral treatment not only for SARS-CoV-2 but maybe for other infections.
Yeah, so Lluís, in this same topic, do you know if there is any initiative for HIV or one of these viruses?
Well, HIV is different, as you know very well. So HIV, it's an integrated provirus. Yeah. It's a retrovirus, it's a lentivirus that is integrated. The problem with HIV, you can silence the expression of the gene, but you still have the genome and inserted somewhere and in different parts, in different cells. So the question for CRISPR there would be to try to remove this HIV genome integrated. But in order to do this, you need to know where this HIV has been integrated, because you want to use neighboring sequences to delete and to promote the deletion. Well, this is still very challenging. It's not impossible, but it's very challenging. I'm saying it's not impossible because a similar experiment was already done in George Church lab with pigs, and this was done by a colleague that you know very well, Marc Well. Marc Well, that is at the UPF here in Barcelona. Marc Well was working in George Church, and they decided to inactivate more than 60 copies of the porcine endogenous retrovirus, the PERVs, and they were successful in doing this. So basically, you can imagine you can target some sequences either neighboring or at the ends of the HIV genome, and you can promote the removal of the genome. This will be actually curing, because for curing an HIV patient at the moment, now we can deal and we can convert a life-threatening disease into a chronic disease with the antiviral treatment, with a cocktail of proteins that are antiviral. But in order to cure for good this patient, either you remove the entire cells of the body and you substitute with a transplant of the hematopoietic cells with free of HIV cells. This is one possibility. This has been done in some cases. If you combine this with some other mutations that prevent the entry of the HIV, such as the CCR5, or the alternative is to develop a CRISPR approach in which you remove by deleting these integrating copies, but both are very challenging.
Yeah. Here you have Marina.
Okay, Marina, Nuria, and Arnau.
Yes.
So Marina says, I would like to know how knowing can have therapeutic benefit in some cases. Oh, yes. This is how non-homologous enjoining can have therapeutic care. Well, I was illustrating this with an example that was published in Cell Stem Cell in 2019 by Paula Rio and Susana Navarro in Juan Bueren's lab in CIEMAT. I really invite you to read this paper. This is a Fanconi anemia treatment. Fanconi anemia, there are proteins that are required for the hematopoietic stem cells to develop. So Fanconi anemia patients, they have a deficit in hematopoiesis, plus they are not prepared to deal with DNA mutations, because Fanconi anemia are part of the repairing system that we have in our body, the Fanconi anemia proteins. So what happens is that they were targeting with CRISPR-Cas some mutations in hematopoietic stem cells from Fanconi anemia patients. These mutations were generating the whole variety of different alleles I told you before. Some of these mutations were converting the initial mutation into something that is a productive protein. For instance, if you're missing one nucleotide and you are deleting two more nucleotides, you are returning back into frame. You know, every 3 nucleotides, it's an amino acid. If you happen to delete 2 additional amino acids, you are returning in frame, and perhaps you are producing a protein that is probably not wild type, but is able to restitute, at least in part, Fanconi anemia protein. And since Fanconi anemia protein are stimulating, are a proliferating parameter, as soon as this happens, even if it happens only in 1 in 10 million cells, this single cell that has restituted the Fanconi anemia will have a proliferative advantage. And in a couple of different cell doublings, you will have in the culture all cells that we will be cultivating will be coming from that particular cells, whereas the rest of cells will no longer proliferate. So this is why you can use this proliferate advantage as a benefit, in this case using non-homologous enjoining. So this was Marina. Nuria. Prime editing, you are extending one of the DNA strands, doing a nick, and with the reverse transcriptase. What happens in the other chain? It's another nick. No. So basically, with the prime editing, remember, basically you are combining different technologies. First, you're no longer using a Cas9, traditional Cas9. So traditional Cas9 opens up the chromosome, cuts double strand. You are not doing this. You are only opening one strand. And by doing this, you are releasing the possibility to, let's say, to pair that DNA strand that you're opening, you can pair it with the extension of the RNA guide, which is now much longer than with the traditional Cas9. And then the 3' of this RNA guide, which is now called PAG RNA, this PEC RNA, that's the one that you can pair with the opposite DNA strand. And because you can pair, and because you have a simultaneous reverse transcriptase domain, you can sustain the generation, the synthesis of new DNA molecule using the RNA as a template. Of course, with prime editing, you cannot do everything. So this has to be clear, and also with base editing. So let's say you have a disease that is caused by a deletion of 2 kb in a given gene. Well, this is not a disease that you can treat with base editing. So you need another setup. You need another way of restoring the missing part. So this is inverted. This is not something you can deal with base editing. You need a CRISPR-Cas9 traditional. Base editing is suitable for those diseases that are associated with point mutations, which, by the way, are the majority of rare disease, and the majority of diseases with a genetic basis, there are point mutations. So this is why it is so. There are so many expectations with the base editing, and prime editing is let's say a peculiarity of the base editing, or let's say the prime editing is another way of correcting few nucleotides. You can correct maybe 4, 5, I think at the maximum 5, 8, 10 nucleotides, but not more. And they need to be at a certain distance to the guide RNA that is positioning this whole complex. This is a bit more challenging, but again, the editors, the prime editors that were presented in 2019, 2 years later, they're already different versions. And the versions that I was presenting in this paper in Genome Biology, it's already obsolete. There is another new version. So basically, we need to be very aware that all these CRISPR-Cas tools, they are evolving very rapidly. And if we are writing a project and we want to use one of these tools, perhaps if we are successful and we are funded and we can do the experiments 1 year later, perhaps what we wrote is no longer true, and perhaps we have to use another tool that has been appearing in the meantime. So it means you need to read a lot. You need to be reading all the different literature, because there are different key labs, and I would say Feng Zhang, David Liu, Eugene Koenig, Kit Young, George Church, all these different labs, they are constantly producing new versions that are always better than the existing ones. So we can never say, I have the best base editor, and that's the one I'm going to be using for the next 5 years. No. Possibly it will be good for the first experiment, but when we have to repeat the experiment, perhaps there is already a better base editors, and we need to be aware and we need to use it. So I think it's
now
ex vivo therapy. It has been shown that the quality of life with monogenic disease can be improved through the use of gene therapy. How far do you think we are to cure complex disease such polygenics and diabetes in the vivo? Well, this is most challenging. So first, I wouldn't call monogenic and polygenic. I would call, let's say, Mendelian disease, monogenic disease, and complex disease. Complex in the meaning that can be caused not only by one but several mutations that could be factors that will promote the development of this disease. Perhaps there are 20 genes that are associated with diabetes. Depending which variants do you have in these 20 genes, your likelihood of developing diabetes over your life will be different. But also, it's not only genetics. Depends on your lifestyle. Depends on what you eat. Depends how much sport you do. Depends on your different treatments or your different drugs, or whether you are exposed to radiation or not, etc. So there are many different factors that are now confluencing, and that they are really making sense for in order to produce some therapeutical benefit. This is why I was underlying this example of lowering cholesterol levels in blood, because this is an example of a complex disease. There are many causes why you can have high levels of cholesterol. So there are genetic causes, but there are dietary causes, and there are many different causes. But all of them, eventually, we know that have very bad consequences, because you will develop a cardiovascular disease, you might develop an atherosclerosis. And what we know is that if we manage to lower the cholesterol, it's going to be good for the patient. So how can we lower the cholesterol no matter what was the origin of this rising in cholesterol levels? And this is why PCSK9 therapy, I think it's a very talented approach how to deal with a complex disease using a modern technology. In this case, using a base editing. We're targeting a gene with base editing. We are interrupting the gene, and this is a repressor. And if we are repressing the repressor, we are promoting what was being repressed. And we are promoting this receptor of these low-density lipoproteins, and this will be captured by the liver cells, and this will result in the reduction of the cholesterol level. Well, this is a clear example how a complex disease can be targeted. You said diabetes. Diabetes still probably still too challenging to be treated with CRISPR, with the exceptions of those where we know the genetic biases. So with the exception of type 1 diabetes, type 2 diabetes, where there is different receptors and different genes involved, plus dietary and lifestyle issues, it's still very challenging. But it doesn't mean that maybe sooner than later can also be treated.
Yeah. So, Lluís, in this discussion, it is essential also to have very good diagnosis tools to clearly define which is the real cause of each disease, or no?
Well, not only this. I mean, we have 20,000 genes, and each of us, we are mutants. So I say this all the time: we are all mutants.
Yeah.
Fortunately, most of our mutations do not express or do not result in something that is compromising our life. In some cases, they do, and this is when you have these pathologies that are being presented. So when you do these genome-wide association studies, these GWAS, then you can realize that, for instance, there are, let's say, 50, 60, 100, or 200 markers in your genome that are associated with the likelihood of developing a given disease. But this is about probabilities. This is not about certainty. So this is something that is very difficult to transmit to patients. And we need to be very careful, those of us that we do genetic diagnosis. When I'm diagnosed as a person with albinism and I have 2 mutations in the tyrosinase, I can know that these mutations will be the cause of that given albinism. I can write a genetic report and I can give this and I can explain this to the patient. But if I detect that I'm carrying a variant that has been associated in a given percentage of people with the development of colon cancer or colorectal cancer, for instance, it doesn't mean exactly that if I'm carrying this variant, I will develop the cancer. And this is something that some people do not understand. And this is why it's so risky and so dangerous to let people use these recreative DNA tests that have been very popular, such as 23andMe or such as other companies, because they will list you a number of variants that will list you your risk of developing a given disease, which doesn't mean that you will develop this disease. Yeah. Because if you are not well educated and you cannot explain what this means, certainty, and what it means, probability, you read you carrying a variant that has been associated in, let's say, 5% or 10% of the cases with patients that eventually develop colorectal cancer, you read this and you think, I'm going to be dying out of colon cancer. And this is not true. So this is why correlating the difference in your genome, in your genome variants with the potential of interacting or interfering or using CRISPR-Cas technology over these genes is a bit problematic. I think we do not yet know enough of these variants in order to take actions, and we need to have some opportunities for treatment of these complex diseases. And again, this PCSK9 for the cardiovascular disease is an example of, we don't care what is the cause, why you are accumulating more cholesterol. What we care is that we can reduce this level of cholesterol. And this is something that was occurring with the cardiovascular disease, but this is not yet, to my knowledge, occurring with diabetes. But maybe sometime we come across a similar development. So
Juan Camilo Ortiz.
Juan Camilo.
For
technology against the antibiotic resistance. Of course, this is some of the projects Francis Mojica is using. So you can target CRISPR-Cas systems in bacteria, shuttle through another bacteria to kill an antibiotic resistant. Why bacteria are resistant to a variety of antibiotics? Because they accumulate plasmids that are carrying proteins that are enabling this bacteria to resist a given antibiotic. If you target that DNA plasmid, and this is something you can do with CRISPR-Cas, and you are using, let's say, bacteriophages to distribute this, you can fight this antibiotic resistance. This is something being explored, yes. Álvaro, do you know if CRISPR technique has already been used in zygotes in order to knock out genetic disease? Well, we're entering in the ethics issues. So I always, during my seminar, I only refer to somatic gene therapy, not to germline gene therapy, right? So you can do in Spain, we have a law that is entitling us, but we have to fulfill a number of requirements. We can use donated embryos coming, I'm assuming that you're talking about human embryos. So we can use donated embryos coming from in vitro fertilization that the parents do no longer need because they have already their children and they have donated these embryos to research. So we can use these embryos and to do experiments in the lab, as long as we're not implanting these embryos in a woman. So as long as we are not converting this into a reproductive use, as long as we are not converting this into a kid. And this is, by the way, what occurred with He Jiankui, these Chinese researchers in Shenzhen, that in the end of 2018, he was using this technology to try to produce a cohort of people that were mutants in the CCR5, hopefully that they would be resistant to HIV. Well, he didn't manage to do this. He created 3 girls, and these 3 girls are there, and they will have to be monitored to the rest of their life. So this is a very dangerous. What has been done in zygotes in Europe? Well, in Europe, we have very few examples. One of these examples is Katinia Khan from Francis Crick Institute in London, in which they use human embryos and compare with mouse embryos, and they realize that OCT4 is a protein that has different functions in 2 mammalian species, in humans and mice. And this is something we would not know if we don't do these experiments. And this is permission that has been given to Ana Veiga here in Barcelona, together with Ángel Raya and Montse Boada. And the 3, they have obtained the first permission for doing this targeting of similar genes that are relevant during the pre-implantation process. And this is an experiment that you can do in the lab. Again, provided that you are not implanting this embryo. And this is something that you can do.
So, yes, Paula is in the same way. I mean, okay. The ethical concerns of gene therapy. You want
okay, so
something
if you are
else.
talking about the ethical concerns, we founded a society that I'm honored to be presiding, that is ARISH. And ARISH is the Association for Responsible Research and Innovation in Gene Editing. It's very easy to find. You type in Google ARISH, ARRIGE.org, and you will learn what can be done regarding the ethical issues on genome editing. Of course, this is about safety and this is about efficacy. When I apply these experiments in my mice, I know that I can select the ones that I want and I can discard the ones that I don't want. But this is something that I can easily do with mice. It's not ethically compatible to do this with patients. I cannot treat 20 patients and pick up only the 2 patients that have responded according to my will and discard the 18 patients. So basically, this is why traditional CRISPR-Cas9 is so prone to variability that probably is not going to be used most of the times in therapies. And this is why I'm stressing. So the take-home message on my seminar today is base editing. It's probably the future of CRISPR, because with base editing, we can be more specific and we can decrease these possibilities of the off-target effects and the possibilities of mosaics that are problematic in human setups. So then we have Claudia.
You have Claudia, that if you can use CRISPR-Cas in more than 1 gene at the same time.
Well, yes. So this is multiplexing CRISPR. Well, again, this has been nicely shown in many different setups. Most of the time, we use 2 different CRISPR in the same gene, and we promote the deletion, but we can promote the deletions of 2, 3, 4, 5, 20,000 genes. The problem is that as soon as you increase the number of genes, you increase the number of double-strand breaks. And the number of double-strand breaks, they are sensed by the cell, and they are interpreted as an insult, as an aggression. And basically, they might decide the cell to senesce. This is a very normal response that some cells, if they realize they cannot handle so many double-strand breaks, they will senesce and they will enter an apoptosis and they will die. So this is something you don't want. So you have to balance the number of genes you want to inactivate with the likelihood that you will be triggering senescence. So when you are targeting genes that are in the same chromosome, even if they are well separated, you have to take into account that perhaps you are also promoting translocations, or you are promoting the removal of whatever it's in the middle. Maybe there are other genes that are relevant. So you have to be very careful when you are multiplexing. On paper, in theory, you can do whatever you want, but in reality, you will be facing consequences. So, Morbuena,
yes.
Are there any other mechanisms in bacteria and archaea that could be applied to modify eukaryotic genome? Well, this will be another 1-hour seminar. So there are many mechanisms. I was telling you at the end, my last slide was this grandmother of this class nucleases, las tatarabuelas, grandmother, that they have been discovered recently. So these are similar nucleases that are triggering double-strand breaks that are tiny. They're smaller than the traditional Cas9. So instead, Cas9 is 1,400 amino acids, whereas some of these grandmother nucleases, they have a few hundred amino acids, and still they do the same. So we don't know how they manage to do the same with so tiny proteins. But this is something that we can inspect and navigate. But I recommend you to read their papers from the NCBI, from Eugene Konin, and from a group of microbiologists in the Weizmann Institute, in which they have discovered dozens of new systems that prokaryotes are using to fight bacteriophages. And we know nothing almost about these new systems. And these systems are totally independent of CRISPR-Cas. They are different. They are using different proteins. They're using different setups. They're using different parameters. And they require somebody to investigate and to elaborate what would be the mechanism. So, in short, there are many mechanisms. And prokaryotes are always an unlimited source of genetic genome editing tools.
You have 2 last questions, Mark.
Mark, what are the effects in gene drive? Well, gene drive, my God. Well, this is, by the way, this is a very good question. But gene drive is another application in which you can bias, for instance, the sex ratio. Normally you have 50% males, 50% females, but you can bias through CRISPR. You can bias the sex towards all males or towards all females, or you can bias that the inheritance of a given trait is not following Mendelian, but non-Mendelian. It means when you're carrying a mutation, you expect 50% of the progeny to inherit that mutation. But with gene drive, if you are carrying a mutation, you can promote 100% of your progeny will be inheriting this mutation. This has some benefits, and these are already being explored to fight this vector-borne disease, such as malaria, Zika, chikungunya, or dengue, or yellow fever. These are all the different diseases that are now being considered using gene drive. You can read a lot about gene drive. We just had different documents on gene drive at the ARISH web page, so I invite you to read there. And Carlos, you talk about different deliveries such as AAVs and nanoparticles. Which of these approaches has the greater future in clinical application? Well, definitely, without any doubt, nanoparticles. So we have to stay away from using viruses. Viruses have been very helpful during a good time. There are many different adeno-associated viruses. There are different serotypes that are targeting different cell types, but they still are viruses. They can trigger immune response. Not everyone can be treated with AAVs. If you have been pre-infected with the same AAV, you will have already antibodies against that particular AAV, and you will not be able to use. Whereas with nanoparticles, you are pretty much free to use. And nanoparticles, if you combine them with these tricks, please pay attention to this transthiritine amyloidosis, in which they combine a nanoparticle and they inserted ApoE in the nanoparticle. And by inserting the ApoE, they also promoted the capture of these nanoparticles by the liver cells. So this is another trick you can use if you want to target nanoparticle to a given cell. And this is something that will be used. And I might be wrong, but to me, I would say that nanoparticles is part of the future. And in half an hour, let me say this, if you want to stay alert, in half an hour at 11:30, we will know the awardees of the new 2021 Nobel Award in Medicine or Physiology. And it is possible that
Lluís, who would you bet on?
Well, if I would be betting Kariko, Weissman, and perhaps Sarah Gilbert, the responsible behind the AstraZeneca adenovirus vaccines, the three people, they have saved millions of human lives because of the vaccination. I don't think there is anything comparable to this. So either they're getting today the medicine award, or on Wednesday, they might be getting the Nobel Award in chemistry. Again, the Swedish Academy of Science and the Karolinska Institute and the Karolinska Academy, they are always very used to make surprises. So maybe there are many more discoveries, but my wish would be that they will be awarding these researchers because their work, not only now, but over 30 years. Katalin Kariko started working on RNA technology in the 90s. And because of this, and this is a very similar parallel history with Francis Mojica and with other pioneers in the CRISPR field, for many years, Kariko and other RNA technologists were forgotten, and they had a hard time to get projects being funded because nobody was believing that RNA could be used as a therapy. Everybody thought that RNA would be a bad idea, and they didn't get projects, they didn't get papers, they even were degraded, so they lost their academic position at the University of Pennsylvania and etc. But look, if you are perseverant enough over the years, at the end, you might be successful. And whenever there was a pandemic and there was an opportunity to test the validity of their proposals, we know this has been coming true. And most of us, including myself, and probably most of you, you have been vaccinated with the RNA technology. And this is why I think that this is probably the best opportunity for being awarded in this Nobel, hopefully. But we don't have to wait much. So in 28 minutes.
A great example. It is a great example, but last year it was a pity that Francis Mojica, he deserves the Nobel Prize.
Yeah, but Francis was the first one to congratulate these two researchers, and he said congratulations, and I'm most glad to have been able to work in a technology that has received the highest distinction, and he was happy with this, and he closed any dispute. So I think we don't have to speak this again.
No, yes. Okay, Lluís, there is one last question by Daniela. It's a particularity of the use of CRISPR lupus, and we close it here.
Okay, so Daniela, so autoimmune disease, as you know, they are very difficult to handle because the immune system is reacting against parts of our body. So basically, here I would say that there are approaches in which you can pretend to alter some of the genes that you know that are behind this autoimmune reaction. If you know which are the genes that are causing this autoimmune reaction, what is the gene encoding a given cytokine, what is the gene encoding a given receptor in the surface of a lymphocyte, you can target that gene with the CRISPR and you can reduce the response, the autoimmune response. Of course, this is not always easy, and this sometimes has consequences, and you might trigger another disease. So this is why you need to do this carefully. But definitely, of course, there are approaches.
Okay, so let's close the session.
Thank
you very much, Lluís, for the outstanding talk, and also for this rich discussion that I'm sure that all the audience, or not only the students, all the audience has enjoyed very much.
Thank you all. Thank you all. And if you still have some more questions, I'm available to email.
Yeah, you can send me the questions to me, and I can direct it to Lluís.
Exactly. Or you can find me in the social networks everywhere. So have a good day, and bye-bye, Josep. Bye-bye.
Okay.
Bye-bye, everyone.
Thank you, and I hope to see you next year.
Bye-bye.
Thank you. See you. Bye-bye. Let's close.